Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we begin Chapter 11, Bioremediation. Microorganisms, plants, and their biological activities can transform, remove, accumulate, or immobilize pollutants in soil and water. But a pollutant disappearing from one measurement does not automatically prove successful cleanup. It may have moved to another compartment, become temporarily unavailable, or changed into a toxic intermediate. Reliable remediation connects pollutant chemistry, site conditions, biological mechanism, transformation products, bioavailability, toxicity, and ecological recovery. By the end, you will be able to explain wastewater treatment and biodegradation, compare in-situ and ex-situ approaches, distinguish biostimulation from bioaugmentation, choose aerobic or anaerobic conditions, evaluate phytoremediation and hydrocarbon cleanup, and design a defensible CBSE answer using controls, spatial sampling, chemical analysis, and realistic endpoints. Our roadmap begins with the meaning of remediation success. We then follow wastewater through physical removal, biological treatment, and polishing or disinfection. We examine how microbial metabolism and commutabolism transform organic pollutants, and why molecular structure, accessibility, nutrients, temperature, pH, and electron acceptors influence the rate. We compare treatment at the contaminated site with excavation and controlled processing elsewhere. Next come biostimulation of indigenous microorganisms, bioaugmentation with introduced degraders, and the different pathways favored by aerobic and anaerobic conditions. We study plant extraction, stabilization, and transformation, as well as microbial attack on oil fractions. Finally, we distinguish degradable organics from metals that cannot be destroyed and construct a monitoring plan using baseline data, controls, spatial replication, transformation product analysis, toxicity tests, and evidence of ecological recovery. The central question separates concentration from risk. A falling parent pollutant concentration may reflect biodegradation, but it may also result from dilution, evaporation, adsorption, or movement beyond the sampling point. Transformation can be beneficial if it produces harmless products, or dangerous if a persistent intermediate accumulates. Immobilization can reduce exposure without destroying the contaminant, so long-term stability matters. A defensible study measures the parent compound, expected intermediates, relevant environmental conditions, and biological effects. It uses untreated or reference locations, baseline measurements, and repeated spatial sampling. Toxicity assays and ecological indicators ask whether organisms are actually less affected. The endpoint must be defined before treatment begins and should combine chemical, biological, and exposure evidence rather than relying on color, odor, or a single laboratory number. Bioremediation deliberately uses organisms, cells, enzymes, or biological interactions to reduce pollutant concentration, mobility, or toxicity. Microorganisms may use an organic contaminant as a carbon or energy source, or transform it incidentally while metabolizing another substrate. The latter is called commutabolism. Plants may take up contaminants, limit their movement around roots, or support degrading microorganisms in the rhizosphere. Biological precipitation or redox change can immobilize metals. Each mechanism creates a different endpoint. Mineralization of an organic pollutant can produce carbon dioxide, water, and inorganic products, while stabilization leaves material at the site but reduces mobility. Therefore, a complete answer states the organism or community, pollutant, reaction or transport mechanism, required site conditions, measured products, and residual risk. Bioremediation is a mechanism-based strategy, not simply the presence of microbes or plants. Wastewater treatment commonly begins with screening, grit removal, and sedimentation to separate large or settleable material. Primary treatment mainly changes the physical load. It does not remove every dissolved pollutant. Secondary treatment uses microbial communities in systems such as activated sludge or biofilms to oxidize biodegradable organic matter and, under suitable conditions, transform nitrogen. Aerobic zones support oxidative pathways, while anoxic or anaerobic zones enable other reactions. Biomass is then separated from the treated water, with some returned when the process requires it, and excess sludge handled safely. Further treatment may remove nutrients or specific contaminants, followed by disinfection before discharge or reuse. Performance is assessed through appropriate chemical, microbiological, and operational measurements. Clear-looking effluent is not sufficient evidence of low organic load, pathogen safety, or ecological suitability. Biodegradation is a sequence of enzyme-catalyzed transformations. The pollutant must first be accessible to the organism or extracellular enzyme. Hydrophobic compounds may sob strongly to soil or form separate phases, lowering apparent availability even when total concentration is high. Molecular structure determines which initial reaction is possible and whether the organism possesses the necessary enzymes. Aerobic degradation commonly begins with oxygen-dependent activation, while anaerobic pathways use different chemistry and electron acceptors. Nutrients support biomass, but excessive addition can disturb the environment. Temperature and pH affect enzyme activity and community growth. Complete mineralization converts organic carbon to simple inorganic products, but partial degradation yields intermediates. Evidence therefore includes loss of the parent, formation and later loss of expected intermediates, changes in electron acceptors or products, microbial activity, and reduced toxicity. In-situ remediation treats soil or groundwater at the contaminated location. It can reduce excavation, transport, and worker exposure, but subsurface heterogeneity may prevent nutrients, oxygen, or organisms from reaching every contaminated zone. Monitoring is difficult when pollutant plumes move. Ex-situ remediation removes soil or pumps water for treatment in engineered systems such as biopiles, land farming, or reactors. This provides greater control of mixing, temperature, moisture, and collection, but excavation and transport increase disturbance, cost, and potential exposure. The best choice depends on pollutant depth and chemistry, site access, geology, time, treatment goals, nearby receptors, and the degree of process control required. A comparison should not claim that one method is universally superior. State the site-specific advantage, limitation, monitoring approach, and residual management plan for each. Biostimulation improves conditions for indigenous microorganisms already capable of useful transformation. Oxygen, an alternative electron acceptor, an electron donor, nutrients, moisture, or pH, may be adjusted after site characterization identifies the limitation. Bioaugmentation introduces a selected strain or consortium with a desired pathway. Introduced organisms must survive, reach the pollutant, compete with the native community, and remain environmentally safe. They may fail if site conditions are unsuitable or the contaminant is inaccessible. A strong experiment compares an untreated control, condition amendment alone, introduced organisms alone, and the combined treatment along with natural attenuation monitoring. This design distinguishes stimulation from augmentation and reveals interaction. Neither strategy substitutes for knowledge of pollutant distribution, redox state, nutrient balance, and transformation products. More microorganisms do not guarantee more remediation. Aerobic microorganisms transfer electrons to oxygen and can rapidly degrade many petroleum hydrocarbons and other organics when oxygen reaches the contamination. Anaerobic communities use electron acceptors such as nitrate, sulfate, carbon dioxide, or oxidized minerals, depending on the pathway and environment. Some chlorinated compounds that resist aerobic attack can undergo reductive transformation under anaerobic conditions, but partial treatment may accumulate harmful intermediates. A sequential strategy may therefore use anaerobic transformation followed by aerobic polishing. Redox potential, dissolved oxygen, electron donors, electron acceptors, parent compounds, and intermediates should be monitored together. Adding oxygen to every site is not automatically correct, and creating reducing conditions can mobilize some metals. Select the redox strategy from contaminant chemistry, existing site conditions, and the complete pathway required to reach a safer endpoint. Phytoremediation uses plants and their associated microorganisms. In phytoextraction, roots take up a contaminant and transfer it into harvestable biomass. This is useful only when uptake, biomass yield, and safe disposal or recovery make sense. Phytostabilization reduces erosion, leaching, or bioavailability without removing the contaminant, so long-term site management is required. Plant enzymes or rhizosphere microbes can transform some organic compounds, while volatilization may transfer material to the atmosphere and must be evaluated carefully. Root depth limits the reachable zone. seasonal growth, climate and soil conditions affect performance. Grazing or food chain transfer must be prevented where accumulated contaminants create risk. Evidence includes soil and plant concentrations, mass balance, leachability, toxicity and vegetation health, not plant survival alone. Crude oil and fuels contain mixtures ranging from relatively biodegradable compounds to heavier persistent fractions. No single microorganism usually degrades every component. Communities divide the metabolic work when oxygen, nutrients, moisture and temperature are suitable. After a spill, evaporation, dissolution, dispersion, sorption, and weathering change both measured concentration and bioavailability. Nutrient addition may stimulate degradation but can also cause ecological imbalance if excessive. Physical recovery and containment often precede or accompany biological treatment. Monitoring should measure representative hydrocarbon fractions and transformation products across space and time. Toxicity tests, microbial activity, and recovery of affected organisms provide biological evidence. A shoreline that looks cleaner may still contain buried or sorbed residues. The cleanup conclusion must match the fractions, depth, and ecological functions actually evaluated. Unlike organic pollutants, elemental metals cannot be mineralized into harmless carbon dioxide and water. Biological processes can accumulate them inside cells or plants, bind them to cell surfaces, precipitate them as less soluble forms, or change their oxidation state and therefore mobility or toxicity. For example, a microbial redox reaction may immobilize one metal under reducing conditions while mobilizing another. Removal requires collection of metal-containing biomass or another separable phase. Immobilization leaves the metal on site, so future changes in pH, oxygen or redox conditions can cause remobilization. Evaluation must measure total concentration, chemical form, leachability, bioavailability and toxicity. A lower dissolved concentration alone may reflect transfer to sediment rather than true removal. Safe handling of concentrated biomass or sludge is part of the remediation plan. Persistent organic pollutants resist degradation because of stable molecular structures, limited solubility, strong sorption, or absence of suitable biological pathways. A specialized microorganism may transform one bond, while another organism or physical-chemical step completes treatment. Cometabolism can initiate transformation when an enzyme produced for another substrate also attacks the pollutant, but the contaminant itself may not support growth. Supplying the primary substrate must therefore be controlled to avoid excessive competing demand or unwanted byproducts. Combined treatment may increase accessibility before biological polishing. Persistence also means that short trials can give misleading conclusions. Long-term monitoring should include parent compound, known and unknown transformation products, bioavailability, toxicity and rebound after treatment stops. Partial disappearance without a mass balance can conceal transfer or accumulation. A monitoring plan begins with baseline mapping of pollutant concentration, depth, groundwater flow, soil or water properties, and biological condition. Sampling locations include treatment zones, untreated controls, upgradient or reference areas, and likely pathways of movement. Replication is spatial, as well as temporal, because contaminated sites are heterogeneous. Chemical measurements track parent compounds, intermediates, and relevant geochemical indicators. Molecular or culture methods can show microbial potential or community change, but gene presence does not prove field activity. Toxicity tests and ecological indicators assess biological consequence. Quality assurance uses blanks, standards, duplicates, and chain of custody records. Sampling continues after active treatment to detect rebound or remobilization. The study should report detection limits, variability, and missing evidence. One convenient sample cannot represent an entire site. A remediation endpoint is the predefined evidence needed to stop or change treatment. A concentration target may be required, but it must apply to relevant depths, media, and exposure pathways. Transformation products should be below safe limits, and the remaining fraction should not be unacceptably bioavailable or mobile. Toxicity should decline in suitable assays, and important ecological functions should show recovery relative to reference conditions. The endpoint must also be stable. Concentrations should not rebound when oxygen addition, feeding, or pumping stops. Secondary impacts such as nutrient pollution, greenhouse gas production, habitat disturbance or introduced organism spread must remain acceptable. When complete removal is impossible, risk management may combine stabilization, containment, and monitoring. A defensible conclusion states what was achieved, what remains, how uncertainty was handled, and what long-term controls continue. Bioremediation is often less disruptive than excavation, but it is not risk-free. Incomplete pathways can accumulate intermediates more toxic or mobile than the parent compound. Nutrient or electron donor addition can alter oxygen, pH, and community structure. Introduced organisms may fail, spread, or exchange genetic material. Plants can move contaminants into food webs, and contaminated biomass must be handled safely. Aerosols, pathogens, flammable hydrocarbons, and toxic chemicals require appropriate worker protection and containment. Treatment can also redistribute pollutants through pumping, volatilization, or erosion. Risk assessment identifies receptors, exposure routes, failure modes, and monitoring triggers before intervention. Adaptive management changes the treatment when evidence shows unexpected behavior. The environmental benefit must exceed the combined risks of contamination, treatment, and residual uncertainty. For a CBSE answer, define bioremediation as biological reduction of pollutant concentration, mobility, or toxicity, then state that disappearance alone is insufficient. Draw wastewater treatment as physical primary treatment, microbial secondary treatment, separation, further treatment or disinfection, and sludge management. Compare in-situ and ex-situ by location, control, disturbance, cost, heterogeneity, and monitoring. Distinguish biostimulation from bioaugmentation and propose untreated, amendment-only, organism-only, and combined controls. For aerobic versus anaerobic treatment, name the terminal electron acceptor and possible intermediate. In phytoremediation, distinguish extraction, stabilization, and transformation, including biomass disposal. State that metals cannot be destroyed. Finish every application with parent and product analysis, bioavailability, toxicity, ecological recovery, rebound, and a realistic limitation. Close your notes and rebuild the chapter. Bioremediation uses microorganisms, plants, or biological components to remove, transform, immobilize, or detoxify pollutants. Wastewater treatment combines physical separation, microbial conversion, and further treatment. Biodegradation depends on pollutant structure, accessibility, enzymes, nutrients, pH, temperature, and electron acceptors. In-situ treatment reduces excavation but offers less control. Ex-situ treatment increases control with greater disturbance. Biostimulation supports indigenous degraders, while bioaugmentation introduces selected organisms. Aerobic and anaerobic pathways use different electron acceptors and may work sequentially. Plants can extract, stabilize, or transform contaminants. Hydrocarbon mixtures require communities and fraction-specific monitoring. Metals cannot be destroyed, and persistent organics may need combined treatment. Baseline mapping controls spatial replication, parent and product analysis, toxicity, ecological recovery, and rebound monitoring support a defensible endpoint.